Hello everyone. In this section, we are going to talk about gluconeogenesis. That's a big word, but by the time we finish today, you are going to understand exactly what it is, why the body needs it, and how it works step by step. I am Brindaronika, working as a tutor in Tribhulam College of Nursing. Before we start, let me tell you what we are going to cover today. By the end of this class, you should be able to define gluconeogenesis, where it happens in the body, list the reactions involved, explain each step with the enzyme responsible, and finally compare it with glycolysis because the two are very closely connected. Okay, so let me ask you something. Imagine you haven't eaten for 8 hours. Maybe you were fasting or you skip a meal. What happens to your blood glucose? It starts to fall, right? Now here's the problem. Your brain and your red blood cells run entirely on glucose. They cannot use fat. They cannot switch to something else. So what does the body do? It makes its own glucose. That process, making new glucose from non-carbohydrate materials, is called gluconeogenesis. Gluco means glucose, neo means new, genesis means creation. New glucose creation. Simple. And it mainly happens in the liver, kicking in during fasting, starvation, or heavy exercise. Okay, now we'll see the definition. Gluconeogenesis is the formation of glucose or glycogen from non-carbohydrate precursors. The key phrase here is non-carbohydrate. The raw materials are not sugars. They are things like amino acids, lactate, glycerol molecules that are not carbohydrate. But the liver takes them and converts them into glucose. We all talk about this precursors in detail shortly. Now, why does this matter? Why does the body even need gluconeogenesis? Let me give you 4 reasons. First, it maintains blood glucose during fasting and starvation. This is critical for the brain and red blood cells, which have no alternative fuel. Without gluconeogenesis, blood sugar would crash dangerously low. Second one is it maintains the levels of intermediates in the citric acid cycle, keeping cellular energy production going. Third one is it acts like a recycling system. When your muscles work hard and produce lactate, the liver picks the lactate from the blood and converts it back into glucose. Glycerol released from fat breakdown also gets recycled the same way. The liver is literally cleaning up metabolic waste and turning it into fuel. And fourth one is this is clinically important. In patients who are critically ill, like those with severe infections or major injuries, gluconeogenesis goes into overdrive. The body overproduces glucose, leading to hyperglycemia, which can actually make the patient worse. You will see this clinical condition, especially in ICU patients. Next is site. Where does the gluconeogenesis happens? Primarily in the liver, to a lesser extent in the kidneys, and more specifically it happens across two compartments, both the cytoplasm and the mitochondria of liver and kidney cells. Now, why only liver and kidney? Because these are the only tissues that contain two specific enzymes. One is glucose 6-phosphate and another one is fructose 1,6 biphosphate. These enzymes are absent in muscle and brain. That's why those organs cannot produce free glucose for export. Next, reactions in gluconeogenesis. Now here is something is really interesting about gluconeogenesis. It does not build an entirely new pathway. It actually borrows most of its steps from glycolysis and runs them in reverse. Think of glycolysis going from glucose down to pyruvate. Gluconeogenesis goes the other way, from pyruvate back up to glucose. But here's the catch. 3 steps in glycolysis are irreversible. You cannot run them backwards. Those 3 steps involve the enzymes hexokinase, phosphofructokinase and pyruvate kinase. So gluconeogenesis has to bypass these three steps using completely different enzyme. Next, enzymes overview. The four unique enzymes that gluconeogenesis uses, the one that do not appear in glycolysis, they are pyruvate carboxylase and phosphonol pyruvate carboxykinase, fructose 1,6 biphosphate and glucose 6 phosphate. These four enzymes handle the 3 bypass point. We are going to go through each one right now in the following slides. Next, precursors. Before the steps, let's quickly identify the starting material, the precursors. First one is lactate. Produced in muscle and red blood cells during anaerobic activity. Remember the burn you feel during intense exercise, that lactate building up. The liver collects it and converts it to glucose. And second one is amino acids, especially alanine. During starvation, the body breaks down muscle protein and sends amino acids to the liver. And next one is glycerol, released when triglycerides, fats are broken down in adipose tissue. And last, pyruvate, the directly entry point of the main pathway. None of these are carbohydrate. All of them can be fed into gluconeogenesis to make glucose. Okay, next we'll move into the very important thing, steps of gluconeogenesis. Step 1, this is the first bypass, converting pyruvate to phosphophenol pyruvate. In glycolysis, pyruvate kinase converts PEP to pyruvate, and that step is irreversible going forward. So gluconeogenesis uses two enzymes to go backward. First, pyruvate carboxylase. This enzyme is found in the mitochondria of liver and kidney cells. It is absent in muscle, which again explains why muscle cannot do gluconeogenesis. This enzyme converts pyruvate into oxaloacetate. It requires ATP, biotin as a cofactor, manganese ions and CO2. And the second one is pep carboxykinase. This enzyme then converts oxaloacetate to phosphophenol pyruvate. So two enzymes to bypass one glycolytic step. That gives you a sense of how thermodynamically unfavorable this direction is. The body needs two separate reactions. There is an important spatial detail here. Pyruvate carboxylase works inside the mitochondria, but the next step of gluconeogenesis happens in the cytosol. So, oxaloacetate produced inside the mitochondria needs to get out. The problem is that oxaloacetate cannot directly cross the mitochondrial membrane, so it gets converted to malate or aspirate. When can cross and is then converted back to oxaloacetate in the cytosol. This shuttle mechanism is an important detail that comes up in the detailed question. And next, step 2, the second bypass. In glycolysis, phosphofructokinase converts fructose 6 phosphate to fructose 1,6 biphosphate. That step is also irreversible. So, in gluconeogenesis, a different enzyme called fructose 1,6 biphosphate does a reverse reaction. It takes fructose 1,6 biphosphate, adds water and removes the phosphate group to give fructose 6 phosphate. This enzyme is present in liver and kidney only, and like the glycolytic step, this reaction also irreversible. It only goes in one direction. The body drives the pathway forward by making each step thermodynamically irreversible. Look at these two reactions side by side. In glycolysis, phosphofructokinase uses ATP to add a phosphate group to fructose 6 phosphate. In gluconeogenesis, fructose 1,6 biphosphate uses water to remove that phosphate group. They do exactly opposite reactions, and they are both regulated in a way that prevents them from running at the same time. Because if they did, you had just be burning ATP in a futile cycle with no net output. The body prevents this through allosteric regulation. Next, we'll see the step 3, the final bypass and the last step of the entire pathway. glycolysis, hexokinase shapes glucose inside the cell by phosphorylating it to glucose 6 phosphate, and that step is irreversible. So for the body to export free glucose into the bloodstream, it uses glucose 6 phosphate, which removes that phosphate group. The product is free glucose, which can now leave the cell and enter the blood. This enzyme is present in liver, kidney and small intestine, but critically it is absent in the brain and muscle. That's why the brain and muscle cannot release glucose into the blood. They can consume it, but they cannot export it. Only the liver can do it. The liver is the glucose factory of the body. Again, the contrast is clean. Hexokinase in glycolysis phosphorylate glucose, trapping it inside the cell. Glucose 6 phosphatase in gluconeogenesis dephosphorylates it, freeing to leave. Two opposite enzymes, two opposite outcomes, and again only the liver has glucose 6 phosphate, which is why only the liver can export glucose to the bloodstream. Take a good look at this diagram. It shows the full pathway from pyruvate all the way up to glucose. You can see the 3 bypass points clearly marked. Trace it from bottom to top: pyruvate, oxaloacetate, phosphophenyl pyruvate. Then through the shared steps with glycolysis, then the second bypass at fructose 1,6 biphosphate, then fructose 6 biphosphate, then glucose 6 phosphate, and finally free glucose. In your exam, if you can draw and label this pathway correctly, you have already secured a significant portion of your marks on this topic. Okay, next we'll see the regulation of gluconeogenesis. How does the body control gluconeogenesis? When does it turn it on? When does it turn it off? So there are 4 levels are there. We'll see one by one. The first level of control is hormonal. Glucagon releasing during fasting when blood glucose is low, and cortisol released during stress. Both enhance gluconeogenesis. They activate the key enzymes and increase gene expression. So insulin released after a meal when blood glucose is high does the opposite. It suppresses gluconeogenesis because there are already plenty of glucose available. So the simple way to remember it: fasting switches gluconeogenesis on, fed state switches it off. And then second regulation is substrate regulation. Substrate availability also plays a role during fasting. Lactate, amino acids and glycerol all rise in the blood, providing more raw material and pushing gluconeogenesis forward. And the third one is allosteric regulation. The molecule fructose 2,6 biphosphate is a key regulator. When blood glucose is high, these molecule levels rise, activating PFK in glycolysis and simultaneously inhibiting gluconeogenesis. When blood glucose drops, its levels fall, removing the inhibition and allowing gluconeogenesis to proceed. It's a beautiful feedback mechanism. And the last regulation is transcriptional regulation. So transcription factors like CIEP, FOX1 and PGC1 alpha control the expression of gluconeogenic genes during prolonged fasting. The body actually increases the production of these enzymes at the gene level, ramping up the cell capacity to produce glucose. This is a long-term adaptation. So we have seen 4 levels of control. One is hormonal, second one is substrate, third one is allosteric, and fourth one is transcriptional, all working together to fine tune this single pathway that tells you how critical it's for survival. Next, very important thing, what is the difference between glycolysis and gluconeogenesis? So glycolysis is a set of reaction that convert glucose into pyruvate molecules. But gluconeogenesis is the production of glucose from non-carbohydrate sources. Then The raw material is a glucose. In gluconeogenesis, raw material is lactate amino acids like alanine and glycerols. Then in glycolysis, it occurs in the cytoplasm, but gluconeogenesis occurs in the cytoplasm and mitochondria. Glycolysis is a catabolic process. Gluconeogenesis is a anabolic process, and the glycolysis is a exonergic reaction. It is a endornic reaction, and glycolysis occurs through 10 reaction, but gluconeogenesis, it occurs to essentially irreversible reactions in the glycolytic pathway are bypassed by 4 new reactions. So this is a difference between the glycolysis and gluconeogenesis. Alright, let's close. Gluconeogenesis is the body's way of manufacturing glucose from non-carbohydrate sources like lactate, pyruvate, glycerol and amino acids to maintain blood sugar during fasting, starvation or intense exercise. It happens mainly in the liver and kidneys across both the mitochondria and cytoplasm. It bypasses 3 irreversible glycolytic steps using 4 unique enzymes, and it's tightly regulated by hormones, substrates, allosteric factors and gene expression. Okay, thank you.